Could scientists be traveling back in time to solve an old problem with photosynthesis? Let's talk. Published in Science on the 15th of March in 2022, the Hanson lab from Cornell University has described an extremely creative means of solving an old problem. This problem has to do with the much admired and much vaunted enzyme RuBisCO. In comparison to other enzymes, it just doesn't turn product over very quickly. And you've probably heard that RuBisCO is the most abundant enzyme on the planet, and one of the reasons why is it's just so terrible. Imagine a business in which you had to hire 500,000 people just to get $1 million in profit. Well, that wouldn't be very efficient, but that's what RuBisCO's like. For each molecule of RuBisCO, it only produces about 2 or 5 product molecules a second. Compare that with carbonic anhydrase that produces 1 million product molecules per second. RuBisCO has got some real evolutionary constraints that hold it back. In this paper, the authors suggest going back to the past to select a RuBisCO enzyme that might work better in our future. So let's talk a little bit about this enzyme RuBisCO. RuBisCO is an acronym for ribulose bisphosphate carboxylase oxidase. It takes a 5-carbon molecule, ribulose 1,5-bisphosphate, and adds carbon dioxide to it, giving it the name carboxylase. It also functions on oxygen too, so it can add oxygen so it functions as an oxidase. So RuBisCO is effectively an enzyme with competing substrates. Ribulose bisphosphate carboxylase oxidase would act on carbon dioxide at some times, performing photosynthesis. At other times, it would act on oxygen, performing photorespiration. Evolution is really acting on 2 aspects of the enzyme. One is the ability to discriminate carbon dioxide from oxygen. Another is the ability to produce a lot of product. And unfortunately for the enzyme and the organisms that contain that enzyme, when the ability to grab lots of carbon dioxide over oxygen increases, the ability to produce a lot of product decreases. So what we're left with is an enzyme that functions in this kind of unhappy medium, and as a result, plants and other photosynthetic organisms need to produce an awful lot of RuBisCO. So people have been trying to improve RuBisCO for some time and have had some problems. There are real technical difficulties, there are other challenges, but basically it's been a difficult time to find the right RuBisCO to make the right changes. So in this paper, the authors describe how they sequence the RuBisCO genes for many organisms, and then they are able to create a family tree, a history of these RuBisCO enzymes. And when they do that, they can infer what the sequences were of older RuBisCO enzymes. So what are these genes? What goes into encoding the RuBisCO enzyme? Well, it turns out there are 2 genes. There's a gene located in the chloroplast, and there's a gene located in the nuclear DNA. So they've isolated the genes for RuBisCO from the tobacco plant. These they're able to then modify in order so that they resemble these ancient RuBisCOs. Those ancient RuBisCOs are then expressed in E. coli. They can then test the kinetic parameters of these enzymes. How well do they work? And what do they find? Well, they find that these enzymes turn over product at a faster rate. They also state that these ancient RuBisCOs have a high selectivity as well. So we see something in these ancient RuBisCOs that we don't in the current RuBisCOs. That is a high selectivity for carbon dioxide and the ability to turn over lots of product. They have yet to try this in vivo. They have yet to introduce these genes into tobacco plants. But of course, that will be the next step, and we'll see in the tobacco plant, will these modified, historical, ancient RuBisCOs be able to perform photosynthesis at a rate faster than the current enzymes? Will they be able to get more biomass out of these tobacco plants? And if it works, they could theoretically use the same technology in many other types of plants that are of economic value. They could sequence, they could determine the evolutionary history of these plants. They can infer the sequences of the RuBisCO genes. They can then make the modifications, put them back into the original plant, and see once again, do these plants have higher growth? Do they produce more fruit? So I hope you enjoyed that little episode of time travel and saw within it the ability of scientists to think extremely creatively. One thing about science that is so underreported and underappreciated is the creative aspect of it. And here we see a great example. People really using their minds to come up with novel solutions. Thank you very much.